Hi, my name is Mr. Easley. Unfortunately, Mr. Anderson couldn't make it to today's podcast, so I'll be taking over. This is Biology Essentials video 93, which is DNA synthesis. So before we get into the details of the actual process of DNA synthesis, I just wanted to give y'all just an idea of the scope of DNA synthesis in humans. So from the cell cycle, there's S phase and interphase, which involves DNA synthesis. And the whole goal of mitosis, at least, is to produce daughter cells that have the same number and type of DNA. And so DNA synthesis is an especially important step. But one thing that the models where we only use 2 chromosomes to represent all the DNA in a cell, it really undermines just how much DNA are in human cells. So if every nucleotide pair, and here's a basic model of DNA with if each nitrogenous base over here was the size of just a letter in a biology textbook, it would take over 1,400 AP Biology, Campbell biology textbooks to just represent all the nucleotides in just one human cell. And so the next question is just naturally, how does the cell synthesize that amount of DNA? Because that is so much. So let's get started. All right, so here's a little guide that I'm going to try to follow to a degree while talking through this podcast. So, of course, we're talking about titular DNA synthesis, but before we actually get into the process, I'm going to talk about some rules that are really helpful to remember. They mostly involve the structure of DNA, and yeah, they're just helpful for actually talking about the process. I'm going to talk about 2 important topics involved in DNA synthesis, which are the leading strand and lagging strand. And then after we finish all of that, I'm going to come over here and talk about mismatch repair and that should say nucleotide excision repair. But there are 2 really important systems that basically involve what the body does when DNA isn't synthesized correctly. All right, so I'm going to talk about a few of those rules I mentioned in the last slide. The first one is that DNA has complementary bases, the little nitrogenous bases over here. This is a basic element of DNA structure. It's just that adenine always matches with thymine, cytosine with guanine, and vice versa. And it's just really important to remember that when we replicate these strands, they have to line up pretty much perfectly. And the second one, this is a bit of a new kind of word, but so DNA runs antiparallel. So in this diagram, this strand runs 5' to 3', and this one runs 3' to 5' from the perspective of that one. That's a bit complicated, so I just kind of colored some paper strips. Basically, this is what antiparallelism is. It's just that they're parallel, but they run in opposite directions. So the red end is on that side, red end's on that side of that one. And that's really, really important as well. And we'll see why later. And then the last one, this is a pretty complicated diagram, but it's just saying that nucleotides can only be added to the 3' end of DNA. So the 3' end is it's not an arbitrary designation of either end of the DNA molecule. It has a reason, but that's exceeds the scope of this video. And so, yeah, just because of the way that the enzymes and polymerases work in DNA, nucleotides can only be added to this 3' end, and this is not a good color. So if you see in this diagram, this is a nucleotide, and these are 2 different steps. So it's getting added over here, and then you can see there's one more nucleotide right there. And this is a very, very important one, even though they're all important. But this especially helps with explaining the concept of the lagging strand. All right, so before we can actually synthesize new DNA here, we have to unwind the parent DNA so we can act on the individual strands, because DNA replication is semi-conservative. We take one part of the original strand, one half of it, one of these strands, and then we build onto it, essentially. And so we need to unwind it, and the enzyme helicase right here, and that's there, it actually unwinds this. And what helicase forms is this thing called a replication fork right there, and it's basically the point right here where helicase actually unzips. It makes a Y shape, and it's kind of the middle region right there. It's just a terminology thing. But the way I like to think about helicase is it's pretty much just unzipping a zipper. And so in a zipper, you have kind of almost complementary little segments, and helicase is just pulling the zipper down, and you separate both parts. And so all the fun enzymes can actually act on it. And so the area that all this unzipping creates is called a replication bubble. And in eukaryotic cells, which we are, there's many replication bubbles because we have many starting points for DNA synthesis. And so that's just some terminology that's pretty important. All right, so before we can build an entire strand, we need a starting point. And so a RNA structure, this isn't mRNA or tRNA, this is just straight RNA. An RNA structure called a primer is going to function as that starting point for us. And sorry that this diagram's kind of complicated because it shows all these enzymes, but it's an extension of the diagram I showed earlier. And another thing I kind of glossed over in the last slide were these things called single-stranded binding proteins. And basically all they do is they keep the replication bubble from collapsing back into the normal strand that helicase tried ever so hard to unzip. But these primers, they just function as a starting point for us to actually build all this DNA. And an enzyme called DNA polymerase 3 does that building. And so the placement of the primers is a pretty important concept to understand. And forgive my poor art skills, but I tried drawing a little diagram. So each of these blue lines is one DNA strand, and this is just a big rectangular replication bubble after helicase did its job. And so we have 5' end over here and the 3' end over here. And remember that DNA runs antiparallel, so our primer is going to be 3' that way and 5' that way. And because we can only build off the 3' end, we can say that DNA is going to be synthesized that way. And because the other strand is also antiparallel, DNA is going to run that way. And so this just kind of shows the importance of the antiparallelism of the two DNA strands. And basically the primers are placed kind of in the middle, but facing a certain way. So if the middle is about there, that one's going to face that way and that one's going to face that way to just make sure that the nucleotides can only be added in the 3' end. And so because we can continuously add nucleotides to this 3' end without breaking it, we can create this really long strand of just straight nucleotides, and that's called the leading strand. And that's one of the two main parts of DNA synthesis I talked about in the little guide at the beginning of the podcast. So these red arrows over here, these are going to be our leading strands, and we built these off of this first primer, and that's why the primer is important. So then your next question would be, how do you build this part? Because this is empty. We need to build it. But you see that part, that goes from the 5' end. We can't just build that way. That would disagree with our rule that we can only build from the 3' end. So how do we do that? All right, and the answer to that question is we can't. Our enzymes, unfortunately, just don't work that way. And so the body does something really clever. So I thought it would be good to go ahead and introduce this diagram. It's similar to my awful MS Paint drawing, but those are the leading strands. And so you might notice these are the empty portions in my last diagram. There's these tiny little strands, and these are called Okazaki fragments. And so we build more primers on this empty segment such that we can build off the 3' end. So we can still follow our rules. But we're just going to build with DNA polymerase 3 all the way down there, and see we're building from the 3' end. And so what the body does, it just attaches a bunch of these primers in succession to form just these Okazaki fragments, and we call this the lagging strand right here. And so we have our leading strand, lagging strand right there. And so, yeah, we use DNA polymerase 3 to build these Okazaki fragments, and so you see that this is the same little DNA part over here. We see that once we've built this one, there's another primer that's placed over there by RNA primase, which is the enzyme that does that, and we use DNA polymerase 3 to start building that way, and we just keep doing that. And then, because we don't really have RNA in our DNA, our next thing would be, how do we get rid of all these primers, and how do we attach all the DNA together? Right, so the way we actually get rid of all these RNA primers to make it just DNA, there's an enzyme for that, as with all things in DNA synthesis. It's DNA polymerase 1, and if we remember, DNA polymerase 3 is what actually builds each individual, attaches each individual little nucleotide over there, and DNA polymerase 1 removes these primers. Well, saying the enzyme does the action is not entirely correct. It catalyzes that action, but anyway. So it removes these primers, and then it replaces them with nucleotides. So what we have then is a bunch of Okazaki fragments that are just DNA, and then we have our leading strand over there. But if you notice, they're not connected. And so we use another enzyme for that called ligase, which is this thing over here. And I think of ligament. It's a connecting body. And also this should say 1, not 2. Apologies about that. But anyway, so this enzyme called ligase connects everything, and so what we have at the end is this really long DNA strand. And at that point, we're done with DNA synthesis. We've synthesized a complementary daughter strand. And so these replication bubbles from earlier that I mentioned, they eventually all just fuse. And so we have like that. And then they just eventually combine, and we get our two big daughter DNA molecules. And another really important thing to think about here is that both the leading strand and lagging strand are synthesized at the same time. They're synthesized simultaneously. The way I portrayed it in this podcast was that I said it in a more like step-by-step thing, but that's really not accurate to how it really works. Basically, there's just a really big complex of enzymes that DNAs really run through. The enzymes don't come down to act on the DNA. The DNA runs through it like the zipper, like the zipper chain, a zipper like of the chain running through a zipper as you go up. And so that's kind of how it works. But the leading strand and lagging strand are synthesized at the same time, and they are also synthesized at the same rate, which is really interesting to think about. So everything finishes at the same time, because you wouldn't want the leading strand and the lagging strand to finish at different times, because then that can mess up a lot of really intricate timing mechanisms in the cell. So now that we've done DNA synthesis, let's talk a bit about mismatch repair and just some repair methods in the DNA. Right, the last thing we're going to talk about in this little podcast is mismatch repair and excision repair. So this whole process we've just described, DNA synthesis, it isn't perfect, unfortunately. The body sometimes makes mistakes. It puts the wrong base there, which is what happened here in this little diagram, in this little just example I did. So we have our base strand is CGC, and the normal complement for this would be GCG, but that's not what happened here. We have an adenine there, and that's not supposed to happen. And so a type of mismatch repair, which is excision repair, involves a nuclease. I couldn't find a good picture of it, unfortunately, but it's right there. It's a nuclease. It's going to come in and literally just take that out. It's going to just degrade it, and so the A isn't there anymore. But now we have a space there. And so DNA polymerase 3 is going to come in and then put the right base there, which is going to be a guanine in this. And another case where nucleotide excision repair would need to be used is in this case. Might not be actually accurate, but it's just an example. So there's these things called thymine dimers, and as seen in this little explanation here, often caused by ultraviolet radiation, sometimes these two thymine molecules next to each other, they bond together. And that's not what we want. We don't want the nitrogenous bases bonding together. Obviously, we want the phosphate background, which I would just imagine this is the phosphate backbone. Did I say background? Backbone bonding together. We want that, but we don't want individual nitrogenous bases bonding together horizontally like that. We only want hydrogen bonds like that. And so this bond right there, it messes up the geometry of the DNA molecule, so we have that little bump there. And so a nuclease comes in, it takes those problematic thymines out, and it replaces them, and so we've done it. So that's DNA synthesis, and I hope that was helpful.